DNA Replication.
Welcome! Today we'll explore the fascinating process of DNA replication using clay models. But first, to the DNA replication. DNA replication is the process where a cell copies its DNA to ensure accurate genetic information is passed on. Let's unzip the DNA first to explain you the single parts of the DNA.
Better.
Now that we have unzipped the DNA double helix structure, we can explain you the single parts of the DNA. So, the DNA is made up of two strands that run in opposite direction. One strand, the leading strand, is the upper one, runs from the 5' end to the 3' end. Whether the lagging strand runs from the 3' end and to the 5' end. In the middle, there are the bases, those things here, and they are represented by the letters C, G, T, and A. C always stays with G and A always stays with T. The unzipping process is carried out by an enzyme called helicase. Of course, this process continues, but let's take a minute to this point. This, what we see here, is called a replication fork. Why? Because it resembles a fork. If you didn't understand this unzipping process well, we have another example for you. You also could think on the unzipping process by using just one of your sweaters. The zipper slider here is the helicase, and these are the bases of the DNA. The helicase comes and splits them. And here we also have the replication fork. Now that we have two separated strands of DNA, we can make a new strand for each one. Let's start off with the leading strand. To copy the leading strand, we need an enzyme called primate. The primate produces small pieces of RNA. Those RNA bases then attach to the bases of the leading strand. These bases together are called the primer, and the primer marks the starting point for the construction of the new strand. Now we say bye-bye primate, as the DNA polymerase comes into play and will finish the rest. Basically, we are now finished with copying the DNA from the leading strand. This sort of replication is called continuous. It's important to note aside that the primus, or otherwise the starting point, goes until here, because it was formed by the primate enzyme. However, from there on, the DNA polymerase did the rest. And now we will focus on the lagging strand. The lagging strand cannot be replicated in the same continuous way like the leading strand because, and we forgot to say that, I'm sorry, the DNA polymerase can only add DNA bases from the 5' end to the 3' end. Somebody might think now, oh well, perfect, from the 5 to the 3. But the strand that will be replicated is vice versa. The 3' end will be here and the 5' end will be here. So it can only go to the 5 to the 3, so it goes this way. And so it can't be replicated like the leading strand where everything was perfect, but at least the beginning stays the same with the primate enzyme. In the lagging strand, we don't need just one starting point or otherwise one primer, but we need many starting points made up by many primate enzymes. So here's one, and let's say here is one. There will be more, but we just show 2 now. It's enough. Perfect. Now we have 2 primers, or otherwise 2 starting points. Of course, there are more starting points, but we couldn't make the lagging strand long 2 meters. And yeah, now we say again bye-bye primates. And the DNA polymerase will add a short row of DNA bases from the 5' end to the 3' end. So from this starting point to the next starting point. And there are also more DNA polymerases, but we just can't see them here. From the end of this primer to the beginning of this primer, so basically the part that was made by the DNA polymerase, is called an Ozeki fragment. And there are many of those Ozeki fragments, only we can't see them here. And this type of replication is called discontinuous. Let's get back to the leading strand. Unfortunately, we are still not finished. Who would not have thought that we need another enzyme called exonuclease? In fact, the primer here can't stay there because it has to be exchanged by copied DNA bases by the DNA polymerase and not by the primate enzyme. So, this exonuclease strips away the primer so that the gap can then be filled by the DNA polymerase. Now the gap that is left is filled by the DNA polymerase with DNA bases. Finally, to seal up the sequence of DNA into 2 continuous double strands, we need another enzyme. But it's neither the DNA polymerase, nor the helicase, nor the exonuclease, nor the primate enzyme, but it's the DNA ligase. You have to keep in mind that the same process with first the exonuclease that removes the primer, and then it is refilled by the DNA polymerase, and then the DNA ligase seals up everything, happens also at the lagging strand, but that would just have cost us 3 more hours of our time. I ask you for your understanding. But now we are truly finished with copying the DNA. DNA replication is described as semi-conservative because each DNA molecule is made up of one old conserved strand of DNA and one new one.
This was our video. We hope you enjoyed it and thank you for watching.